Good morning. Today I'm going to discuss on plasma proteins and immunoglobulins. Learning objectives are to discuss the major classes of plasma proteins and their principal function, and to discuss the types, structure, functions of immunoglobulins. Three major types: albumin, globulin, and fibrinogen. The normal values are total protein 6.8 to 8 grams percent, albumin 3.5 to 6 gram percent, globulin 2.5 to 3.5 gram percent, fibrinogen 200 to 400 milligram percent. Ag ratio is 1.2 is to 1 to 2.5 is to 1. Synthesis of plasma protein. All albumins and fibrinogens are synthesized by liver only. Similarly, 50 to 80% of globulins is formed in the liver. Remaining amount of globulins is formed entirely into the lymphoid tissues. The Ag ratio can be altered in various liver diseases. Plasma protein can be separated by salting out method, which is the outdated one. Then electrophoresis, ultracentrifugation, and immunoelectrophoresis. This is the electrophoretic separation of plasma protein. The pattern is something like this. The huge band of albumin followed by alpha 1, alpha 2 globulin, beta globulins, and gamma globulins. These are the major classes of plasma protein, their function and diagnostic importance. So, in the pre-albumin category, two proteins come: the TBPA, that is thyroxine binding prealbumin, and retinol binding prealbumin. Principal function is transport of hormone thyroxine, and RBPA transports the vitamin A. Diagnostic importance lies in liver diseases, thyrotoxicosis, and malnutrition. Albumin, it exerts the colloidal osmotic pressure and transports function. Its diagnostic importance is in malnutrition, liver, kidney, and gastrointestinal diseases and malignancies. Then A1 globulin. So various type of alpha 1 globulins are there. RBP, that is retinol binding protein. So it transport vitamin A, while A1 phytoproteins functions are unknown, but its diagnostic importance lies in neural tube defects and also act as a tumor marker. Then alpha 1 protease inhibitor or alpha 1 antitrypsin, that is AAT. It acts as an anti-protease and a natural inhibitor of proteolytic enzyme elastase. API deficiency lies in the lung disorders. Then A1 acid glycoprotein, its function is in tissue repair. In inflammatory diseases and malignant neoplasm, there is a diagnostic importance of AAG. Then prothrombin, its main function is in blood clotting. Diagnostic importance lies in coagulation screen and also as a liver function test. Alpha 2 globulin, there are four types: ceruloplasmin, haptoglobulin, thyroxine bonding protein, alpha 2 macroglobulin. So ceruloplasmin transport the copper. Its importance is in Wilson's disease. Haptoglobin, conservation of iron by binding free hemoglobin. Its importance is in hemolytic disorders. Thyroxine binding protein, it transports thyroxine hormone, and its importance is in thyroid diseases. Alpha 2 macroglobulin, AMG, it acts as a natural anti-protease inhibitor, inhibits the thrombin, trypsin, and pepsin. Its diagnostic importance lies in myeloma, peptic ulcer, nephrotic syndrome, cirrhosis, and collagen disease. Then beta globulins, hemopexin, transferrin, C-reactive protein, and beta 2 macroglobulin. Hemopexin binds heme and prevents the loss of iron. Its diagnostic importance is in hemolytic disorders. Transferrin transports the iron, and importance lies in iron deficiency. C-reactive protein, it is important for body's defense mechanism, while its diagnostic importance is in the non-specific test that may be used instead of ESR. It's a major marker of inflammation, well-established marker of various categories of inflammations. Then beta 2 microglobulin, body's defense mechanism, uh, and importance lies in monitoring myeloma, renal failure. Then gamma globulin. So there are five types: IgA, IgAG, IgM, IgD, and IgE. Their function is in body's defense mechanism in various diseases like liver diseases, infection, autoimmune diseases, and paraproteinemia. Their diagnostic importance is apparent. Fibrinogen, blood coagulation. Now I have a quick look onto the important points regarding the plasma protein. The most abundant plasma protein is albumin, least abundant is alpha-1 globulin. The plasma protein with fastest electrophoretic mobility is pre-albumin, transerythritins followed by albumin. Plasma protein with the slowest electrophoretic motility is gamma globulin. The plasma protein not synthesized in the liver is immunoglobulins. The plasma protein not a glycoprotein, that is albumin. Plasma protein exclusively synthesized in the liver, that is also albumin. So albumin is a globular protein with 585 amino acid residues accounting for approximately 50% of plasma protein. Albumin is synthesized exclusively by the liver. Major important functions played by the albumin are in maintenance of plasma oncotic pressure, transport of metabolite like fatty acids, bilirubin, calcium, certain steroid hormone, copper, some of the plasma, tryptophan, a variety of drugs like sulfonamides, penicillin G, dicumarol, aspirin, digoxin, all of them being transported by albumin. Buffering action, beside it also participates or contribute to the buffering function and maintenance of various body fluids pH system. So albumin has maximum buffering capacity. Nutritive function also it performs. Degradation of albumin provides so many essential amino acids during the malnutrition. Hypoalbuminemia, its clinical significance lies in the hypoalbuminemia, in malnutrition, nephrotic syndrome, cirrhosis of liver. So there is lack of albumin. So normal concentration is less than its concentration is less than the normal value. In hyperalbuminemia, acute dehydration have no in acute dehydration it may happen and have no clinical significance, and it's a reversible one. You can have a lot of intake of water, it will recur. Analbuminemia less than 1 gram per liter, normal is 3.5 to 6 gram per dl. So analbuminemia is a rare hereditary abnormalities that may be there may not be any symptoms or sign, not even edema, due to compensatory increase in plasma globulin concentration. While in the hypoalbuminemia, there is a lot of edema present all through the body, or bipedal edema is very marked. Causes of hypoalbuminemia, it can be physiological or pathological. So physiological can be the pregnancy related, pathological due to the impaired synthesis of malnutrition, malabsorption, chronic liver disease. In all these three things, there is an impaired synthesis of albumin. Increased catabolism is another cause, that is due to the injury, major surgery or trauma, infection, fever, or malignant diseases. Abnormal distribution, severe burns, edema, and ascites. Abnormal or excessive loss is in nephrotic syndrome, gastrointestinal tract disease, burn or certain skin disease, hemorrhage, inflammation. Overhydration due to hemodilution. You can see it here. This is the blood vessel, and this is the extravascular fluid. So During edema, what happens? The albumin content, edema lowers the albumin constant. So what lot of water excuses out into the extravascular places. Globulin constitutes several functions. Lot many fractions of globulins are alpha 1 globulin, alpha 2 globulin, beta globulin, and gamma globulins. So immunoglobulins are gamma globulins, also called the antibodies, constitute about 20% of all the plasma protein produced by plasma cells and by the lymphocytes in response to variety of antigen. This is the schematic presentation of immunoglobulin. You can see it does consist of, this is the common structure of all the immunoglobulin. The basic monomer consists of this blue colored heavy chain. Two heavy chains are there and two light chain. And this is this upside FAB region. This is lower side FC region. Both the chains are joined with each other via disulfide bonds. You can see it here. And this is the papain enzyme cleavage site. I'll tell you shortly about papain enzyme. Then antigen binding site. This FAB region contains the antigen binding sites. Then FC region has two domains, CH2 and CH3 domains. And while FAB region has CH1 domain in the heavy chain. The basic immunoglobulin is a Y-shaped, consists of four polypeptide chains, the two H chains and two L chains. H for heavy, L for light. The four chains are linked by disulfide bond. The L chain may be either two types, kappa or lambda. Similarly, the heavy chain also alpha type, gamma, delta, mu, or epsilon type. Immunoglobulins are named as per their heavy chain content. So different types of immunoglobulins have different types of chains. Like in IgG, it is gamma, IgA, it is alpha. In IgM, it is mu. IgD contains the delta heavy chain. IgE contains the epsilon as in heavy chain. So L and H chains are subdivided into the variable and constant region. L chain consists of one variable and one constant domain. Most of the H chain consists of variable VH and three constant domains. I have already showed you here. I can show you again. The hinge region between the CH1 and CH2 domains confers flexibility and allow both FAB arms to move independently, thus helping them to bind to antigenic site. And the light chain types, light chain may be either two types, kappa or lambda. In a given immunoglobulin, either two kappa or two L lambda chains, but not the mixture of kappa and lambda. Most abundant light chain is kappa type. So enzyme digestion splits, enzyme that is papain. Digestion splits the immunoglobulin molecule into the two fragments, FAB fragments, the fragment for antigen binding located into the variable region, then FC, it lies into the constant region. It is crystallizable fragment or fragment for complement binding. Now, various types of immunoglobulins and their characteristics and function at a glance. Immunoglobulin IgG, it is a monomer. The main antibody in secondary response, it is the only immunoglobulin which crosses the placenta and is the only maternal antibody which protects the fetus. It neutralizes the bacterial toxins and viruses, opsonizes bacteria, making them easier to phagocytize, activates the complements which enhances bacterial killing. IgA, it lies 20% of all the immunoglobulins, major component of cholesterol, also occurs in saliva, tears, in respiratory, intestinal, and genital tracts, genital secretions, sorry. The secretory IgA prevents the attachment of bacteria and viruses to the mucous membrane and helps protect the mucous surface from antigenic attack. It prevents the access of foreign substances to the circulation. Then IgM, it forms 8 to 10% contribution to all the immunoglobulins, the main antibody in the primary response to an antigen and produced by fetus, activates the complement, promotes the phagocytosis and causes lysis of antigenic cells. Antigen receptors on the surface of B lymphocyte. It is IgD is less than 1% labile molecule, and these facts have made the study of IgD function very difficult. So there is uncertainty regard to the function. Then IgE trace amount it is found in trace amounts. It's around 0.004%. Bind mast cells and basophils and leads to rupture of cell membrane, degranulation, and release of histamine. Anti-allergic and anti-parasitic, mediates the immediate hypersensitivity by causing release of histamine from the mast cell and basophils upon exposure to antigen. Defends against the worm infection by causing release of enzymes from eosinophils. Main host defends against the parasitic infection like helminthes, provides protections in the disease osteostomasis. So there are various arrangement how the monomer can be in the dimeric, pentameric form. So this is the structure of monomer. This is dimer. Two monomers are attached via juncture, that is the J chain, proteinase chain. Secretory IgA is a dimer, so it lies in this particular form. Both monomers are attached via J chain. This is the biggest immunoglobulin, have 5 pentamerics. 5, it's a pentamer, and you can see the 5 immunoglobulins are attached, monomers are attached via J chain. Most common immunoglobulin in serum is immunoglobulin G. Least common immunoglobulin in serum is IgE. The largest one is the IgM. The pentamer is IgM. Dimer is IgA. Ig fixes the complement IgG and IgM. Ig present in the secretion. That is immunoglobulin A. Immunoglobulin can cross the placenta, that is IgG. Cannot cross the placenta, IgG to 21G, involved in primary immune response, that is IgM. Ig secondary immunoglobulin secondary immune response, that is IgG. Ig with J chain, that is IgM, and IgA act as a linker of various monomers. Ig with secretory pieces, that is your IgAs. Disorders due to the changes in immunoglobulins. So quantitative changes have been observed in the amount of immunoglobulin in various in the plasma and urine in various pathological conditions like in multiple myeloma, amyloidosis, Benzon's protein, cryoglobulinemia, Waldenstrom microglobulinemia.